These are in listen-only mode.
Welcome everyone to a webinar today on statistics for companion diagnostics. I'm very pleased that we've got three presenters today, Peter Cooper from QIAGEN, Mei-Jean Lee from FDA, and Rachel Hodge from AstraZeneca. I'm Rachel Lawrence. I'm going to chair the meeting as this is a subject that I'm very interested in and have been involved with myself as well. So I'm going to hand over to start the webinar to Peter Cooper. Thank you.
Thanks, Rachel, and thanks to the Scientific Subcommittee for our Scientific Committee for organising today's event. I'm Pete Cooper. I'm Head of Statistics at QIAGEN, and I'm going to take you through an overview of the role of statistics from the companion diagnostics perspective. So it's going to be I'm going to cover the whole development process from initial feasibility studies for the assay through to clinical validation and co-approval of the companion diagnostic and drug. So the presentation is in three sections that you can see there on the slide. And there are two main objectives of this presentation. One is to give you an overview of the whole diagnostic development process. And secondly, to lead into the next two presentations, which focus on the end of the process, which is the clinical validation stage, and in particular the next two presentations focus on clinical efficacy bridging studies. So the definition and uses here are taken from the FDA guidance, and I guess many of you listening will be familiar with the definition. There are many potential uses of an IVD. The 4 PMA approvals that QIAGEN have gained with our partners so far were all in the third bullet there, selecting patients who are eligible for a particular therapeutic treatment. So it's a tightly regulated environment. Generally, companion diagnostics are class 3 devices. There's regulation from the FDA, the first bullet there, and also there's a lot of specific guidance on the design and analysis of specific studies from CLSI, the Clinical and Laboratory Standards Institute. There's also regulation in Japan as well. So one thing to bear in mind in terms of the companion diagnostic is it's not just the assay or the actual test that calls whether or not the mutation is present or not that we're interested in. We also have to bear in mind the pre-analytical side and also the instrumentation and software. So, for example, we need to consider sample collection, sample storage, DNA extraction, and also conduct of the use of the assay where the tubes go into the PCR instrument. So there's a diagram there showing the EGFR workflow at the bottom, and also there's a diagram showing the 5 main areas that we're interested in. And so as statisticians at a diagnostics company, we do get involved in all stages and also in the whole workflow. So there are a lot of interesting challenges in terms of matching up experimental objectives to the instruments configuration. So here we have a model project for companion diagnostics, and we can see the drug program, the diagnostic program, and the regulatory programs all running in parallel with the aim of achieving a simultaneous approval between the companion diagnostic using the PMA route and the drug with an NDA. In this plan, everything's planned really in an organized way. So we can see in the regulatory program that the diagnostic is gaining an IDE in time for the phase 3 trial. So we'll be able to use the diagnostic in the phase 3 trial for co-approval. So in this case, we'll avoid the need for a bridging study. In the diagnostics program, swim lane there in the middle, the diagnostics development splits into two areas, development and verification and validation. And underneath those two boxes, we can see some arrows which break down those areas into the individual studies or individual areas, and we'll go through those briefly in a minute. So in terms of an overview of the statistics role, we have a very broad-ranging role throughout the company, and we do the usual things that you'd expect statisticians to be doing in terms of project inputs, design, analysis, development and methodology. One of the key things we like to do is train the R&D scientists around QIAGEN Group. So the graphic there is a optimal study design graphic, which was a part of a teaser campaign for some training for the scientists. Obviously in companion diagnostics, like everyone else really, we do work with a lot of people, and externally as part of a companion diagnostic co-submission, we'll typically work with a broad project team both here and at the pharma partner, and also with maybe multiple CROs who are aligned alongside the pharma partner. Inside the company, technology is moving fast as ever, and so we have a close relationship with the new tech office and also NGS and NGS bioinformatics. And we also get involved with manufacturing operations on things like sampling inspection and quality control. So what I'd like to do now is take you through just at a very high level the different stages of development to give a feel for that. So firstly, within the development stage, we have assay design. And so here we're looking to develop the reaction mix for the assay, make sure we identify a robust assay which has good precision and is not particularly variable. We obviously need to check the feasibility of the assay, and in particular in the left-hand graphic there, check that we have an assay that will meet the product requirements and make sure that the wild type or mutant negative samples are not going to be falsely called positive. And at the same time, we need to ensure we have an LOD, a limit of detection, of a sufficiently small percentage mutant allele fraction. The right-hand plot shows the relationship between precision and accuracy, and this is from a simulation, obviously making many assumptions in that particular simulation, but it shows a general pattern that as the precision of the standard deviation for the assay, the precision of the assay, as that increases, then quite clearly the percentage misclassified patients will grow. And for any particular distribution of patients in relation to the cut point, we can conduct a simulation and understand what impact any lack of precision is having on misclassification. And then the second stage of development, we call that assay robustness. And here we'll be ensuring a robust assay by looking at the response profiling for different combinations of the key factors in the reaction mix. And we'll be looking to position those factors on plateaus so that any minor variations in those through manufacturing does not impact greatly on the results of the assay. And the left-hand plot gives a pictorial version of a central composite design with just 3 factors. And then the final stage of development is where we set the assay performance characteristics. And you can see in the list there that there are quite a number of those, limit of blank, limit of detection, precision, and so on. And so we'll conduct either individual studies or possibly combine 1 or 2 more characteristics in a single study to establish performance. And these performance characteristics need to be determined in such a way that they are suitable for verification in the verification stage of development. At the same time as conducting all the performance studies, we'll be involved with the manufacturing side and other members of the project team in terms of developing the positive controls and finalising on the reference method we're going to use for determining analytical sensitivity and specificity. And the plot on this slide is just showing a classic LOD plot where we've tested a sample at different concentrations to understand where the LOD is for this particular assay. So that very quickly completes the development stage of the process of developing the companion diagnostic. And now we move on to verification and validation. So for the verification studies, as I say, we now need to check all these performance characteristics that have been set. In particular, we need to verify the product specifications and we need to verify the user needs. In this case, the example plot corresponds to a linearity study, showing that we have a linear assay across a DNA input range, which is really just meaning that our assay will be robust to different concentrations of sample. And then the final stage of verification and validation is the clinical validation stage. And so this is where the remaining part of this talk and the next two talks sit. So clinical validation consists of a clinical accuracy study where we'll be looking at the sensitivity and specificity of clinical samples from an appropriate population, and the phase 3 co-study. So if the CDX is available in time for the start of the phase 3 co-study, then we don't need to do bridging. But if it isn't, then we will have a clinical trial assay. So that's the assay that was used in the original pivotal trial. And we'll have the CDX, the companion diagnostic, that has been tested on samples that were kept. And so we have some typical bridging study tables here where we're looking at the agreement between the CTA and CDX. And we're looking at agreement both unadjusted in terms of the actual samples we have, and also following enrichment adjustment. And so we may need to adjust for enrichment, which will often happen if the samples available for testing with the CDX are in some way not representative of either the percent mutant positive for the study, or possibly also for the percent mutant positive for the underlying intended use population. So just a few further slides to introduce bridging studies. Obviously, if the CDX is used from the start, we don't need a bridging study. We have the CDX used throughout. There's no missing data for it, and we use the study for concurrent approval of both the therapeutic and device. And for this simple graphic, we've got the three main types of study design: non-comparative, comparative select mutation positive only, and comparative enroll all, and stratify by mutation positive or mutation negative. However, if the CTA, the clinical trial assay, is used in the study and the CDX is not used in the study, then we need to retain samples for CDX testing, and we'll need a bridging study to establish efficacy of the therapeutic when using the CDX. And obviously there are a few variations on this theme. It could be part way through that the CDX becomes available, and in this case it's really quite similar to the previous slide. We'd still need to conduct bridging to go from the to ensure we take account of all patients in our efficacy analysis. Alternatively, we might have a position where we have used the CDX in the clinical trial, but the CDX there was a CDX for tissue samples and was used on the actual tissue. Whereas we may also be interested in using a diagnostic for testing mutations present within blood plasma, and in this case we may need to bridge from the tissue to the plasma. So finally, some key considerations, and I note that the other two presentations also have considerations that overlap, so I won't dwell too long on these. But the main things I'd like to emphasize here are that it's really important to have integrated planning between the pharma partner and the diagnostics company. And also it's very important for the pharma partner to consider diagnostic requirements right from the off in terms of the project plan. Because obviously to have the diagnostic ready for the phase 3 study means that there is a considerable amount of work required before we can actually gain an IDE and use the diagnostic in the study. So integrated planning is very important. Obviously the quality of the clinical trial assay is very important too, as a poor assay could dilute results. Timing of availability of CDX is really important. And I think the final 3 bullets are worth emphasizing. So that's recording of sample testing and mutation results information, to really pay a lot of attention to that and make sure that it's complete. We want to minimize missing samples so that we can have a robust bridging study and minimize or completely eradicate any potential issues with selection bias of samples for CDX testing. And it's also really important to pay as much attention to the screen fails as it is to the randomized patients. So for some pivotal studies, the screen fails are in some ways not quite as important as the randomized patients. However, for a bridging study, they are really important and we need to have information on them. So in terms of consent, sample collection, patient baseline data, sample baseline data, it's all very important for the screen fails. In terms of the main analyses in a bridging study, they fall into 3 areas. There's the concordance and enrichment analysis, where we'll look at measures of agreement and adjust those for any enrichment, as I said. We'll conduct representative analysis to illustrate that there's not been any issues with bias that have crept in through availability of non-representative samples in some way. And there are a few different sections to that analysis that you can see within the box on the slide there. And then there's a clinical efficacy bridging analysis. And there are 2 or 3 key things to bear in mind there. One thing is we need to understand, we need to take account of the potential CTA negative, CDX positive patients. Obviously for a selection trial design, we won't actually have clinical outcomes on those patients, so we'll need to make assumptions. To understand the impact those patients may have on the final efficacy, we need to bring the enrichment adjusted PPA into the analysis. And then the other main point is to take account of the missing CDX test results, so we may conduct multiple imputation to ensure the full patient set is included in the analysis. But the next talk by Meijuan Li is going to focus on this area. So following the whole development process and including the clinical validation, we then reach a stage where we can submit for co-approval at the same time, the PMA going to CDRH and the NDA to CEDA. And if everything's gone according to plan, then we'll gain PMA and NDA approval at more or less the same time. And so throughout this process, statistics obviously has a multifaceted role, many different inputs, looking not only at assays but also at instrumentation, also looking at complete workflows, and working with our partners throughout, and also including joint working during the clinical bridging stage as well, if that's required. So thanks very much for your attention, and I'll now hand over to Meijuan.
Thank you, Peter. If anybody has got any questions during the webinar just before measuring starts, feel free to enter them on the webinar chat questions function, and we can we'll be able to take questions that way because everybody's lines are muted. But yeah, if you have got questions and they're directed to a particular speaker, make that clear, or general questions, we should have time for them either after measurings or at the end of the presentations. Thank you. I'll hand over to Mei-Jing now. Thank you.
Thank you for organizer for inviting me, and it's my pleasure to discuss with you today about bridging companion diagnostic device bridging study. And also I think I would like to thank Peter already give a nice introduction and example on bridging study. So I will focus on the bridging study design and the statistical methodology and the challenges we have been seen in the those bridging study. So based on the FDA regulation, there are two different types of companion diagnostic device. First one is called predictive test. So sometimes we use test and the device exchangeable. So in my talk, sometimes I refer to device, sometimes I refer to test. For companion diagnostic device, first one we call the predictive test. This type of a companion diagnostic device, or ACDx, predicts differential treatment effect on the outcome, for example, survival time. This type of device identifies patients who are most likely to benefit from a particular therapeutic product, for example, drug. It also could identify patients who are likely to be at increased risk for serious adverse reaction as a result of a treatment. So in order to demonstrate this device is actually a predictive test, we typically recommend the sponsor to provide clinically and statistically significant treatment by device interaction. In order to do so, both marker positive and marker negative defined by ACDx are required to be enrolled into the clinical trial. The second type of test we call selection test. So for this type of test, the device identify patient or select a patient for treatment such as drug treatment. In clinical validation study, typically only marker positive patients are enrolled. So clinical validation is typically not designed to predict the differential treatment effect on the outcome, because as I mentioned early on, for the predictive device, if you you have to demonstrate the treatment by device interaction. Since most of the time for selection tests, only marker positive are enrolled, so we do not have any information on marker negative population. So we cannot demonstrate. There's no way to demonstrate the differential treatment effect. For this type of device, we recommend sponsors to demonstrate clinically and statistically significant treatment in the selected patient group defined by the device, for example, marker positive. So in my talk, I will focus on the selection test and the selected patient by the device, I assume it to be marker positive. So before I talk about the bridging study design, I just want to talk a little bit about what is CTA and the ACDx, because those notation will be used in my talk. So CTA stands for clinical trial assay. This type of assay could be, for example, early version of companion diagnostic device. This assay is used in clinical trial for the patient marker status determination. However, this device may not or have not been approved or cleared by FDA as a companion diagnostic device. So that's called a CTA. CDx is a device that the sponsor intended to put on the market. So it is a device that's on the FDA regulation or pre-market review for corresponding therapeutic product. So assumption here is that CDx and CTA have the same study in the clinical trial inclusion-exclusion criteria. If they have different inclusion criteria, for example, CTA are talking about women over age 40, but the CDx are talking about general population, there's no age limitation, then that's different inclusion exclusion criteria. So those CTA and the CDx, in order to bridging, they should have same inclusion exclusion criteria. Now people may ask why I already use the CTA, why I want to put the CDx on the market, why I want to use CTA, not the CDx. There's different reasons for CTA being used instead of CDx. I listed two possible reasons here. One is that CDx is not available at the time of the pivotal clinical trial, and the CTA is already available. Second possibility is that CDx, it is available, however, it's not available all the lab where the sample may be collected. So it's maybe only available in the central lab. However, CTA is available at all local lab where samples are collected. So in order to, save the time during, sample shifting from local lab to the central lab, and the sponsor may use the CTA for at those local lab for the marker status determination. So there's, of course, there are other reasons, but that's mainly two reasons for CTA being used instead of CDx in clinical trial patient enrollment for marker status determination. So there are different type of, there are different way of drug and device validation, and they can use different study design. We always talk about drug and device co-development. However, there's always possibility that CDx and the drug may not be validated exact way. So here I listed 6 different type of clinical validation of a companion diagnostic device. I just divided into entire clinical trial into, for example, 3 different study group, just for purpose of my discussion. So suppose I have group 1, group 2, group 3 for first design, and I use the blue marker for blue marker indication that at the enrollment, the patient marker status is determined by CDx. So for design 1, there's no bridging study involved because everybody marker status is determined by CDx. For the second study design, in my late talk, I will call the partial bridging. So the study group 1 is enrolled based on the CTA marker status. So for this group, we have to retest by, the patient sample have to be retested by CDx so that the patient marker status can be determined by CDx. For the group 2 and 3, the marker status are actually determined by CDx. What happens is that in the middle of clinical trial, the CDx may become available. So then we sponsor switch from CTA to CDx. So for this study design, group 2 and 3, there's no bridging study, so only partial bridging study is needed. Now the focus on my talk today is actually study design 3. I use the term for bridging study. Basically, the entire clinical trial study patient and all initially tested by CTA So for all those groups, ideally have to be retested by CDX to determine their marker status based on CDX. Then study 4, 5, 6 are very complicated. Basically involve double bridging because you have two different versions of CTA. And we typically do not recommend use this study design because every time you do the bridging, you basically lose some efficient, and so it's not a very efficient design. So my next the slides in my next few minutes talk, I will focus on the study design 3, which is a full bridging study. Next slide, please. So let's first talk about the definition, although I kind of already stated that. So bridging study definition, this is the agreement between the CDX and the CTA with the samples of the patient enrolled in the clinical trial when CTA is used for patient enrollment in clinical trial. Now I will have to point out that there's another type of agreement study is we so-called follow-on device. I don't have time to talk about that today. But that type of study is you typically use a sample outside of a clinical trial. It's not inside of a clinical trial. So that's the difference. So in the bridging study, the clinical sample is from inside of a clinical trial. That's the key points. So the purpose of the bridging study, first we can assess agreement between the CTA and the CDX. Second, allow us to bridge off the CTA clinical data to CDX. Most importantly, it allows us directly to estimate CDX effectiveness defined by treatment efficacy in its intended patient population. So again, the assumption that bridging study is consistent with the eligible criteria, same as for CDX intended user population. I already mentioned earlier on about CTA and the CDX enrollment criteria. It's the same thing here. So how can we do that? So we will retest all available screening patient sample of clinical trial. So including some of CTA negative, ideally all the enrolled clinical trial CTA positive patient should be retested by CDX. So we will apply both CTA and the CDX to additional patient sample outside clinical trial if necessary. So this is a little bit tricky. I mentioned earlier on, we encourage you to include inside the clinical trial sample, but sometimes if there's an issue with, especially with a negative sample, with missing data, missing sample, not sufficient enough sample, so we occasionally allow that to use outside, but there's a lot of issues. So I don't have time to discuss those issues today, but I just want to point out. So next slide, I would like to talk about different type of a bridging study. I already kind of briefly mentioned different type of bridging study, but again, there's a full bridging study, like the study design, then partial bridging study, basically in the middle of the clinical trial, the assay switch from CTA to CDX. Double bridging study, basically there are two different versions of CTA being used for patient enrollment. And then the bridging will be very complicated. From, for example, you may need to bridging from CTA1 to CTA2 for first part of the study, and then from CTA2 to CDX for the second part of the study, then bridging from CTA1 to CDX use both part 1 and part 2 study. So it is very complicated and not efficient way to do it. So we do not recommend this approach in general. If possible, avoid this type of design. So So I'm going to talk about the full bridging study. Because I used the selection test as an example in my talk, and just again, what is selection test? So for this type of trial, typically only mark possible patients are enrolled. So if you look at the right column in the clinical trial, see I only highlighted that if patient are CTA, they will be enrolled. But if they are CTA positive, they will be enrolled. But if they are CTA negative, those patients are not enrolled. So in the full bridging study, the CTA positive, ideally, all CTA positive enrolled patients will be retested by CDX. But there's two possibilities this may not happen, at least two possibilities. For example, the specimen may not be available because during CTA test stage, all the specimen collected from patient are used up. There's nothing left. So that's one possibility that for those patients, even though enrolled in the clinical trial, but they cannot be retested by CDX. Second possibility that they may have some left over, but the quality of those specimens are not good enough. So even you tested, but they may have no CDX results. So that's the issue for the missing data I think Peter mentioned on. So that's a CTA positive. For CTA negative, typically CDX positive, the prevalence is not like very high. So you may not have to retest every CTA negative by CDX. So it's probably only a proportion of CTA negative patient will be retested by CDX. And the one I highlighted in the red are the ones that do not have CDX results. So because the CTA and the CDX may not perfectly agreed, it's possible that CTA negative could have CDX positive. It's also possible that the CTA positive could have CDX negative. So that's a discordant results between CTA and CDX. So in the next slide, I'm going to talk about the definition about agreement between CTA and CDX. So CDX and CTA depends on which is the results you're conditional on. There's two sets of agreement. First set of agreement we called PPA and NPA. So I denote as Q1 and Q0. Second results we typically call the PPV and NPV. So this is conditional on the CDX. So in my talk, I denote as P1 and P0. Then if in the study, we could estimate what is the proportion of the enrolled or screened patient, the proportion of CTA positive. In my talk, I denote as 5. Then use 5 and Q1 and Q0. Based on base theorem, we can get estimate for P1 and P0, which is PPV and NPV for CDX. The P1 particularly is very important because this metric will be used in the drug efficacy estimates. So that's the set of agreement. Now the next slide, I'm going to talk about the estimation. So I already mentioned the layout for the bridging study. So basically you use in this table, you use the highlight for cell to estimate PPA in NPA. Then based on PPA, NPA, or PPA, NPA, I denote as Q1, Q0. Then based on this and the 5, which is the proportion of positive CTA patients, then you can get a PPV and NPV. So the next page, next slide, I'm going to talk about how to I provided the formula about the PPV, PPA, MPA, and the PPV, MPV. So I mentioned that you can estimate phi from screen clinical trial population. So if you have a previous table, you just plug in this formula to get the estimate of phi and also Q1 and Q0. And the confidence interval will be very straightforward, and you can use the score method to estimate. Now, once you have those three quantity estimates, you could easily estimate P1 and P0. Use the formula I put here. And the formula for the confidence, the method for the confidence interval for the P0, P1, it's a little bit complicated. Of course, there are different ways, but in my slides, I use this delta method. So this is the formula you can use, and if you want to find the detail how this formula is derived, and you could refer to my paper. And like I said, there's other method you can use, both point estimate and the confidence for P1 and P0. So now next so we already talked about agreement between CTA and the CDX. So now fundamental question, the main purpose, I guess, for the bridging study is to what is the drug efficacy in the CDX population? That's the fundamental question. So basically the agreement we discussed previously should be used in the efficacy estimate. So here's a formula. I think there's a lot of definition, but you can, if you're interested, could refer to my paper. I want to point out three important quantity. So P1. So look at the highlight blue formula here, delta 1. What is delta 1? Delta 1 is the treatment efficacy in CDX population. So for example, log hazard ratio for the CDX positive population. And delta 11 is a drug efficacy in double positive. Double means that CTA positive and CDX positive. Delta 01 is a drug efficacy in the CTA negative but CDX positive patients. Now P1 I already mentioned in previous slides. P1 is PPV. So you could use linear combination of delta 11, delta 01 to estimate delta 1. So next slide. Now the problem here is that I mentioned early on, because in typical selection test clinical trial design, CTA patient, CTA negative patient are not enrolled. So we cannot estimate the delta Z1 because they do not have clinical data at all. So that's why I said this data is missing. And also because for CTA positive, for the CTA negative patient, for CTA positive, CDX negative patient, even though they have clinical trial because they are enrolled, however, those are not CDX positive intended user population, so those patients are excluded. So that's why I earlier mentioned about inefficient. That's also part of inefficient. If you have discordant patient between CTA and CTX, CTA and CDX, some patients even enrolled, but they are not part of the CDX intended user population, so they will be excluded. Now, another layer of complex is that for some patients, even they are enrolled clinical trial. However, because they are not retested by CDX, so we do not know their status. So those patients we also cannot be used in estimated CDX drug efficacy. So those also are excluded. So that's another inefficient in terms of bridging study. And the next slide, I'm just I mentioned the formula, so here just plug in what I just described in early on. So delta 1, you have delta 1, 1, you can estimate from the clinical trial for the double positive patient. P1, you could estimate from bridging study based on PPA and NPA and then convert to PPV. And so you just, and the delta 01 is a tricky part, as I mentioned, because we do not have data. So for this one, you could assume, for example, this value is, for example, if you talk about log hazard ratio, or hazard ratio, then you could say this is some way between delta 11, which the efficacy is a double positive, and if you're assuming there's no effect at all, that hazard ratio will be 1. So something like that to give you basic sensitivity analysis. So what I presented here is just one possibility of estimating those drug efficacy. Of course, I just want to mention there's different way to do that, but because time constraint, I just give you one approach. You could refer to my paper for more detail. So in next few slides, I just want to briefly talk about the issue in bridging study. So CTA negative are not properly documented. Important covariates are collected for CTA negative patient. Total number of CTA negative screening sample may not be collected recorded. And so that's for if that's the case, then the phi, which is the probability of CTA positive, cannot be correctly estimated. Consequently, the treatment efficacy in CDX positive delta 1, which is cannot be correctly estimated. CTA patient, negative patient may not be consented to be retested by CDX. That's another possibility. You do not have CDX results, and the CTA may not be early version of CDX. For example, CTA and CDX may be very different. So then the agreement between the two assays could be low, and also CTA could be not just one assay, could be multiple assays. That's even more complicated. Then valid CDX results may not be possible for enrolled patients CTA positive due to sample stability, quality, volume, or patient consent issues. So those are issues for bridging study. And next page, please. So good practice for bridging study. We recommend that the analysis plan in the study design is completely pre-specified, ideal before you study pivotal clinical trial. And I can't maximize maximum number of screening clinical trials sample, if not all, including at least some CTA negative sample from the pivotal clinical trial. And those samples should meet the bridging study eligibility criteria. No selection bias should be introduced, and a large portion of random pivotal clinical trial, if not all sample, should be included in the bridging study. And ideal quality of the archived specimen for retesting by CDX is acceptable, and the quantity of the specimen is sufficient. So that way to avoid missing data. And also baseline covariates and other relevant variables are available, especially most of the time has issue with CTA negative patients. Those patients we usually do not have same set of covariates in criteria. Although I understand that it's probably difficult to do, but I encourage that to correct all covariates as much as possible, even for CTA negative patients. And the clinical data in the CTA test results should be masked to the test operator or investigator. Of course, ideally, you should power both bridging study in a clinical trial. And for targeted device performance, power and type 1 error rate, etc. So should be considered in a bridging study design. And the missing data issue can cause a lot of issue, and I already mentioned like a data set for pivotal clinical trial, for example, for CTA positive set, and may not be entire all set being available to be retested by CDx. And the CDx evaluable set, a subset of CTA positive, which are not available for retesting by CDx, or results are not acceptable. So, so it's possible that the randomized treatment assignment is because it's performing on CTA positive set. So it's possible that within CDx positive set may not be balanced. And the data set used to for estimating drug efficacy on CDx patient a group which is double positive one is a subset of a CDx evaluable subset. So identify a set of covariates which can affect both test results. So for example, you may use logistic regression or linear model to test whether which covariates could impact on test results, and check for imbalance in the covariates between the sample in CDx evaluable set and the CDx non-evaluable set. And the check also please check the clinical outcome between the both the CDx positive, CDx evaluable set and the CDx non-evaluable set or unevaluable set. And conduct complete case analysis and also conduct some sensitivity analysis on the different assumption. And I mentioned early on about the how do you do the delta 0-1 because those cannot estimate directly from clinical trial. And of course, the last point will be discuss important differences among the various approach. And I already mentioned about the possible issue for randomization because CDx double positive is just a subset of CTA positive, and the randomization of a treatment assignment may not be maintained because you only maybe a small, especially if you have a lot of missing data. So check the imbalance in the covariates between the sample in treatment and the control, between those treatment and control group. And use appropriate statistical method to adjust the covariates when you estimate the drug efficacy in the CTA and the CDx double positive group. And here's my concluding remarks. So design the bridging study in the clinical trial carefully, and the key points is to plan ahead of time. Please specify data analysis plan and the study design, archive maximum number of screened trial sample, and eliminate potential bias at the design stage. And of course, if possible, address bias issue at the data analysis stage, like the enrichment, missing data, non-randomization issue I mentioned, and collect all the baseline covariates and power the study. Next page. And the bridging study can be complicated to design, and a complicated design to analyze in many statistical issues I mentioned earlier on. And the CTA measurement performance often not a well characterized assay and could diluting the drug efficacy, may lead to a larger sized clinical trial. So you could refer my paper, and I cited here. And the personalized medicine is actually a total medical device. It's a drug marker device patient, and use good well characterize the CDx and avoid bridging study if possible. And we encourage you to discuss with us and at the design stage. Thank you, and please let me know if you have any questions.
Thank you very much, Mijing. I have had one question from one of the attendees, so I'll just read it out. So the question was, what is the relative importance of demonstrating drug efficacy in the bridging study versus showing a high level of agreement between the CTA and the CDX?
What I didn't catch the first part of your statement. You are saying that how to demonstrate drug efficacy?
Yeah.
Oh, okay.
No, what's the relative importance of demonstrating, you know, which is more important, sort of demonstrating the drug efficacy or showing a high level of agreement?
Oh, okay, good question. So if you have very high agreements and if you have less missing, small proportion, only very small proportion of data are missing, and if drug efficacy in CTA population is high, then for such a case, high agreement will equal to high drug efficacy in CDX population. However, if you have, even you have high agreements, if you have missing data, if original CTA drug efficacy are not great, then we will be very concerned because the drug efficacy in the CDX population will not be meet clinical or statistical significant acceptance criteria. So in the short, I would say CDX pass-through patient drug efficacy is more important.
Okay, thank you very much. I'm now going to hand over to Rachel Hodge from AstraZeneca, who's going to give us a case study example of a bridging study. Thank you.
Thank you, Rachel, and thank you to the previous speakers. I believe that leaves a nice introduction and covers some of the topics on my presentation now. As Peter alluded to, obviously the clinical development and the diagnostic development is generally done in collaboration, and the work I'm about to briefly go through was done in collaboration with Roche Molecular Systems, and so they're just acknowledged as a collaboration between AstraZeneca and RMS. So I'm going to briefly introduce the drug that we were testing this on in development, as well as the study design and the patient selection, and go over some tissue results, how we use the local tissue results in the actual study recruitment and analysis versus wanting to obtain a central tissue result and approval of that central tissue test. And then the analysis of efficacy that was conducted on the local versus that central test. And we also looked at bridging between a tissue test and a plasma test, and can run why that was important to the patients, and again look at the analysis of the concordance and the analysis of the efficacy. And then I'll briefly touch on the issues considering which I know Peter and Meiji have briefly talked about on the missing data and particularly on the screen failures. Development was irreversible duplicate cell, a TKI79M mutation. So essentially we required to develop a diagnostic tool that would be able to reliably detect the presence of a T79M mutation in either tissue and plasma for the patients to see if they were eligible to hopefully receive a new treatment. We conducted a study. It was a global phase 2 open label single arm study to assess the safety and efficacy of osimertinib in a patient population who had non-small cell lung cancer. It was second line, they had progressed on previous therapy, and whose tumour specimens demonstrated the T79M positive result. It was a single arm study and the primary endpoint was overall response rate by a blinded independent central review. And this was defined as the number or proportion of patients having at least one visit response of a complete or partial response that was confirmed at least 4 weeks later. The study screened 472 patients and of those 383 provided pathology, so tissue data specimens, and 210 of those were then entered into the study and received at least one dose of study drug. Patients could continue to receive osimertinib until objective disease progression, or for as long as they were receiving clinical benefit from the treatment. And this is actually one of 2 studies that was used to support the accelerated approval of osimertinib. So briefly touching on the statistics used to assess the effectiveness of the new tool. We have looked here at the positive percentage agreement, the negative percentage agreement, and the overall percentage agreement. And here we're defining the positive percentage agreement as the number of true positives. So essentially the number of patients that were identified as being T79M positive on the test, so under the new diagnostic tool, and were also positive under the gold standard test. So in our case, this was a test that was already approved. And that's divided by, again, the total number of true positives plus the number of false positives. And here the number of false positives would be the number of patients who were identified under that new test as having a positive T79M test result, when actually the gold standard test identified that patient as being T79M negative. The negative percentage agreement is defined as the number of true negatives. So that's the number of patients who had a negative T79M result by both tests, divided by the number of true negatives plus the number of false negatives. So the false negatives is defined as the new test making a negative prediction when the gold standard test defines that patient as being T79M positive. I briefly touched on the overall percentage agreement, and the formula is shown on the screen, but here this doesn't differentiate between the agreement on the positives and negatives, and so our primary focus was on the PPA and the NPA. So looking first at the concordance between the two central tests. So here version 1 was the approved test, and version 2 was the test that we were wishing to have approved as the diagnostic tool to identify the mutation in these patients. And so we conducted a bridging study, a bridging analysis to perform and establish the effectiveness of the version 2 test through retrospective testing of patients who had been enrolled into the study and provided a sample which was tested under version 1. And here we can see that we've calculated the PPA and the NPA as defined on the previous slide. And here we can see that we achieve 96% of the results that were identified as positive under both tests, as of the number that were positive plus the false positive result, as being high, showing a good level of concordance between both versions of the tests. The NPA was also high, up again at 96, just over 96%. So here we were seeing high concordance and good reliability of the version 2 of the test versus version 1 for identification of the T79M mutation in our screened patients. So then we moved on to look at what efficacy data we had, and we replicated the primary analysis, so overall response rate for the patients who were then positive for the tissue T79M mutation in version 2 of the test. So here we can see that under the patients recruited into the study, so those that were received at least one dose of drug, so the 210 patients, of those patients that came in and were therefore T79M positive under version 1, 128 of those were defined as a responder, so a 61% response rate. And repeating that analysis based on those patients who were T79M positive under version 2, so here we had 204 subjects, we achieved a response rate of just over 62% here. The analysis was then repeated as a sensitivity on the investigator assessment, and here we can see between version 1 and version 2 of the test there was a very high overall response agreement between both of those. So moving now to consider the tissue versus the plasma concordance. As a company, we were very interested in exploring whether we could have a plasma test approved, because it's less invasive than a tissue biopsy, and therefore it would be in the best interest of patients. And some patients may not be well enough or eligible to have a tissue biopsy, and therefore it was allowing those patients to have the opportunity of having a test which may identify them as being T79M positive, and therefore allowing them to be eligible for treatment. So first of all, we then had to consider how many patients provided us with the plasma sample, of which we had matching tissue specimens, and we were fortunate here that 90% of the screening patients also provided us with a plasma sample which was available for testing. So the first thing we did here is again compare the tissue data, which is in the columns, with the plasma data, the plasma T79M result, which is in the rows. And here we can see that the PPA and the NPA are approximately 60% and 80%. And these are the results that exclude the invalid and the no plasma sample. So essentially the PPA of 58.7% is coming from the 131 in the COBAS version 1 test positive cell and the plasma test version 2 positive cell out of 223. So out of the total in the COBAS test 1 T79M positive, excluding the invalid and the no plasma sample from 10 patients. And this essentially shows that the plasma test itself is good at identifying those truly negative cases, but maybe it's not quite as good as it could be, and it may actually miss identifying cases that were truly positive. And that's reflected in the PPA being around 60%, maybe than a more preferred percentage of being up at sort of the 80 or 90%. So we also conducted the primary endpoint analysis again for the overall response rate by the plasma test. So to note here that actually obviously the patients there where we had the efficacy data would have been all patients that came into the study and received at least one dose. And so actually the plasma test results, we do need to remember that these patients were then obviously positive for the T79M mutation under version 1 of the test. But here we can see that the plasma positive patients had a consistent overall response rate with that observed across the study. And actually those patients that were T79M negative by the plasma test also had a good overall response rate, which may then be overestimated potentially by the proportion of those patients who were identified as being plasma T79M negative as actually being positive due to the PPA being around 60%. So this essentially left us with the question of what about the efficacy in the cohort of patients who were T79M plasma negative, plasma positive, but tissue negative. And obviously we didn't observe this data. These patients didn't come into our study, but essentially it was of interest because these patients could be available or eligible for treatment if we were to have approval of the plasma test. So here we have the situation where we were then trying to estimate efficacy from a non-treated population. So no efficacy was observed, so we had to model an observed efficacy under a range of assumptions. And here we assumed that the T79M negative patients had a proportion of the efficacy of that observed in the positive patients. And here we used a range from 80% down to 0. So no efficacy at all in those patients that were T79M negative. We used a weighted average based on the proportions observed from the PPA, and on the screen now we can see that for example, the 48.8% is 80% of the efficacy observed of the 61% across the cohorts, and weighting that by the PPA, if we were to then see all patients treated by the plasma test being T79M positive, we were estimating an overall response rate of 59%. And you can see how, when varying the range of efficacy in that T79M negative population from 48.8%, so 80% of that efficacy, down to 0, then leads to a range of overall response rates of 59% down to 51%. The overall response rates presented here were considered robust in patients who were their T79M positive by the plasma test. So some of the problems we've heard a little bit about, and I won't dwell on for too long, is that you may want to think about when setting up the study, is actually that you don't always get all the data you need from the screening patients. So where I presented data on the PPA and the NPA and the concordance between having approval for a diagnostic test, say between a version 1 and a version 2, or even between tissue and plasma, it's important that we are able to collect those plasma samples for patients that are screened, and not just those that are randomized or entered into the study. And that's because we can look at all the screen data there, and that gives us a larger sample size for looking at that concordance. In our studies, I mean, I think we were fortunate in having 90% of patients provide a plasma sample as well as a tissue sample. But in other studies, that may have been an issue, and our actual protocol did not require a plasma sample to be provided for eligibility into the study. And again, just to dwell on the fact that the plasma data itself was not used, or the plasma test was not used for recruitment for eligibility into the study. And so we had no efficacy data in the plasma positive tissue negative patients, and therefore we had to use a range of assumptions to estimate the efficacy for approval of the plasma test. So you would have seen that when we looked at that concordance, they were on all screened patients. And so again, it's just important to consider when designing the study and when setting up the protocol of achieving that data, ideally in those screening patients. We also did have to look for the submission at the summary of demographics, and those were also shown to show consistency. And so the baseline characteristics for those screen failures were also important here. And whilst that data isn't always collected, or maybe even always cleaned to the same level as the treated patients, if you are looking at doing some diagnostic analysis, that would be one other consideration that would be useful to add. So moving to my conclusion slide, essentially additional considerations are needed when you're developing a diagnostic tool. So collection of that demographic data, as well as hopefully tissue and plasma samples in our situation from the screen failure patients, was really important. And it definitely aided us having a positive review by the CDRH at the time of the tissue and the plasma submission. We actually also required parallel submissions, because once osimertinib was hopefully approved, obviously patients would need to have the availability of having a test to identify if they were T79M positive. So actually this work was conducted in parallel to the clinical submission. And then just to note, because of the slightly lower PPA from the plasma result when comparing to the tissue, the actual approved plasma label indicates that if a patient's tissue, if a patient's plasma result comes back as T79M negative, it does allow for that patient to be followed up by a tissue test to see if they are truly negative or if that was a false negative identification from the plasma test. And I think that's key to allow patients who have a need for a new drug to still have access without relying on just the plasma test. But again, it was important to have that plasma test approved because it's an easier option and much less invasive for the patients than a tissue test. So thank you for that. I will hand now back over to Rachel Lawrence for any questions.
Thank you very much, Rachel, and for all the presenters for their presentations today. And it was really nice to hear that sort of case study highlighting some of the things that both Peter and Mei-Jing had talked about. We've had a couple of questions so far. If anyone else has got some, feel free to type them in the questions box now. We'll answer these couple of questions first. So the question which was to Mei-Jing to answer for us: how do you evaluate PPA and PPV when there's NPV when there is not a gold standard assay? So for example, in the case of a new assay.
Hello, this is Mei-Jing. So the PPA NPA or PPV NPV concordance between CTA and CDX. So basically we compare the results where the CTA, for example, corpuscle, and the CDX corpuscle also. That we say they agree to each other. Of course, it's depending on which compared to which. That's why one set we call it PPA NPA, another set we call it PPV NPV. So for both those sets of agreement, we actually do not compare to the reference standard. So there's no reference standard involved. I hope that answers your question.
Thank you. And another question we had was, how would you say we can rule out false positives or negative or false negative results in the setting of companion diagnostics?
So I can start, and other on the panelists can jump in, and if I miss anything. So this is more. This is a very good question, and this is more like for assay accuracy issues. So in the assay development stage, you really have to make sure that your assay is accurate and precise, because you call negative. For example, the true marker status for given patient is true marker positive. Test may call you, CDX may call you negative. There's two issues this could happen. One is that your assay is not accurate. Second is that your assay is not precise. So you really have to, in the developer stage, you really have to make sure your assay is very accurate and precise. Another thing, in addition to those accuracy and the precision, the cutoff, how you set up a cutoff also is important, because, for example, you true cutoff, which is differentiate marker positive from the negative. If, for example, if you set it too high, then some marker positive patient you may call negative. So you have to make sure that your cutoff is correctly and set. And of course, those accuracy and the cutoff and the precision should be all figured out at the design stage. Once the clinical trial started, no way you can change it, because typically with the company you lock everything at the time your validation starts. So everything, those issue about false positive and the false negative You have to make sure you fix those at the developer stage.
Okay, thank you. Do any of the other panelists have anything to add to that point? Okay,
Yeah, Rachel,
thanks.
this is Peter.
Okay.
I'd just say that
Peter,
obviously, as Mejan said, at the design stage, we're very careful to design the assay so that we don't get false negatives, and that we understand what the limit of detection is very well, and that we have a precise assay. And then right through the development, verification and validation stages, we do build a lot of evidence to demonstrate the performance of the assay. So that happens right from the very start through development, verification and validation. And of course there's usually two sizable studies in terms of sensitivity and specificity, in terms of the analytical sensitivity and specificity, and then the clinical validation as well, looking at sensitivity and specificity again. So by the time it comes to submission, there is a lot of evidence around the performance of the assay.
Great, thank you very much. We've just got time very quickly. There was a follow-up question to the to the previous question, which I'll read out. Although I might just need to ask the for a slight clarification, because the question is, what if there is no CDX and you're validating a new assay, and is the comparison with the clinical outcome enough when a CDX is not available? I'm a little confused by the question myself, because you know, we're talking about in the context of approval of a CDX. So I'm wondering if the person posing the question it is meaning that in the context, or if you're talking about perhaps in a more an exploratory way in terms of looking at concordance of different types of, perhaps different types of diagnostic testing. If any of the panelists have a better idea where that question might be heading, if you could comment. Meijing.
Yeah, I can comment on. So I think I think your question may be, I already have CTA. Why I need to do CDX at all? As Rachel pointed out, CTA actually is not the device sponsor wants to put on the market. So yes, CTA could be perfect, but the patient, when they get the results, it's about CDX. So I should know what is CDX results, what CDX performance look like. Is that right? So that's the purpose of the bridging study. We need to know the device which will be on the market, and how the device performance is like. The CTA is not the assay we are interested in this type of clinical trial validation. It's like an intermediate step. CTA is like an intermediate step. So we need to know the final step, which is the CDX device performance. I hope this answers your question.
Yeah, thank you. So if anyone does have any further questions or want some clarification, feel free to get in touch with myself, and I can put you in touch with the presenters if you have anything else. Also, just to say, the full recording of the webinar will be available on the PSI YouTube channel shortly after the end of this meeting, if you want to listen to it, or if you've got any colleagues who weren't able to make it and might be interested. So thank you. Right.
So
Go ahead.
Question.
Oh, yeah.
And so there's another question. Say, is comparison with the clinical outcome enough when CDX is not available?
Yeah, could you comment on that? Yeah.
Yeah, so still I'm not exactly sure what the question is about, because you are talking about comparison CTA to the clinical outcome. because if CDX is not available, then I'm not sure what's the purpose of the whole study, because the bridging study, clinical validation study, and it is about CDX. That's the device the sponsor are interested to put on the market. It's nothing else, nothing about the CTA. It is about the CDX. So if it's CDX not available, then we have no such submission. So if you're talking about CTA, then if the sponsor interested in the CTA, then CTA should be the assay put on the market. Then we no longer call CTA. It's actually we'll call that is proposed companion diagnostic device. So when we call CTA, basically it's just intermediate step assay, which are not sponsors are not interested to put on the market at all. So to answer your question, no, it's not sufficient. We do need to look at CDX results. And if CDX is not available, then there's no bridging study. There's no purpose of such a study at all.
Okay, thank you very much. Thank you to all the presenters today. I just had a final slide showing future PSI committee scientific meetings that are coming up in 2017 for anybody who's interested in any of the sort of topics. So you can see these on their website. So thank you very much for joining today. I hope you've found it useful and informative, and we look forward to you attending any of our future PSI meetings. Thank you very much.